Nowadays, in corona and the critical situations we are in, we should stay at home and save our lives until we can produce a vaccine. So, on another hand, we have another problems, such as economic recession and starvation. So, we need to provide food sources specifically in poultry and save the birds from invading another diseases by using genome editing techniques. One of these techniques is CRISPR-Cas9, which stands for cluster regulatory interspaced palindromic repeats. Today I will talk about applications of CRISPR-Cas9 in poultry. In my outlines, CRISPR-Cas9, methods of CRISPR-Cas9, genome editing, and the CRISPR strategies of CRISPR-Cas9, applications in quail or chicken, strengths and limitations of CRISPR-Cas9, and conclusion. First, what is the CRISPR-Cas9? CRISPR-Cas9 is one of genomic editing techniques that was adapted from naturally during genome editing system in bacteria. Cas9 nucleases can be directed by short RNAs to introduce bases cleavage at endogenous genomic loci. We have three methods of CRISPR-Cas9. Firstly, CRISPR-Cas9 can be directed by short RNAs or converted into editing enzyme to facilitate homology-directed repair. And third method, multiple guide sequences can be also encoded into a single CRISPR array. Genome editing and CRISPR-Cas9. First, they invented mega nucleases, then zinc finger nucleases, continued by TALEN, which stands for transcriptional activator like vector nucleases. Then our topic today, CRISPR-Cas9. We have three strategies of delivery of CRISPR-Cas9. The easiest one is using same vector to express Cas9 protein and small gated RNA protein. And the second one, introduce a mixture of the Cas9 mRNA and the small guide RNA that be translated in the cell. And the third strategy, directly deliver into cells a mixture of the Cas9 protein and the small guide RNA that have been translated. Applications of CRISPR-Cas9 in poultry. CRISPR technique enables us to understand the function of genes and or modify the animal phenotype to fit a specific scientific or production target. First application is application of CRISPR-Cas9 in quail. Scientists have been modified primordial germ cell in vitro and injected into the blood vessel of the egg. Then adenoviral CRISPR-Cas9 vector optimized for expression of guide RNA. And Cas9 gene in avian cells was injected into the quail placental in newly egg that resulted in mutated chimers have desired traits. Applications of CRISPR-Cas9 in chicken. As we know, the first study using a CRISPR-Cas9 system in chickens was published in 2015 and involved the electroporation of chicken embryos with plasmids encoding Cas9 and guide RNAs against the transcription factor PA67. First application we will talk about in chicken is tank binding kinase. Tank binding kinase is an enzyme with kinase activity. Scientists noticed that the gene related with this enzyme limit the mutation rate. By using effective gene knockout system that in a chicken embryonic fibroblast, they obtain new cell lines have increasing rate of mutation. Now we will talk about using CRISPR in protein manipulation and antiviral infection. To reproduce antiviral chicken cell line, scientists using chicken NHE1 gene, which have a physiological role in mammals, which regulate the intracellular pH and cytoskeletal organization. They drive primordial germ cells for blood aspiration of male imperious receptor in third line for the chicken NHE1 allele and susceptible to avian necrosis infection virus. Then introduce into chicken HNE1 that make a residue in tryptophan amino acid that play a critical role in virus entry that make the chicken antiviral necrosis virus. In avian species, to establish valuable chicken lines with highly productive protein source for humans, we can modify protein genes such as ovalbumin and overtransferrin that presented in egg, that differs in nutrient level, allergenicity, or the protection of biofunctional material from another egg proteins, and can be also development of disease-resistant avian line for pandemic diseases such as avian influenza and avian necrosis virus that have caused enormous damage to poultry industry. The scientists have just suggested that overexpression of a small herpin RNA, specifically regulation viral RNA polymerase, that may be prevent avian influenza or avian necrosis virus. Now we can discuss strengths and limitations of CRISPR-Cas9. Firstly, strengths. CRISPR-Cas9 can modify DNA with greater precision than existing technologies and don't need to pair with several cleaving enzymes, and also can be used to target multiple genes. Its limitations can effect of target can alter the function of a gene and may result in genomic instability. Conclusion. Transgenic poultry play an effective role in economic enhancement and feeding population that can be solve the starvation and poverty problems by increasing the production using editing genome technology. This is my topic reference, and thank you.